Hello, everyone. I hope you are doing well today. Got a video. We'll talk about some of the physiological processes of the grapevine. So let's get started here. So here I got a couple pictures of veraison kind of happening. This was just taken 2 days ago. Um, so you can see some coloring up of the berries here. Still a lot of green ones on this cluster. This cluster has much more purple berries starting over here as well. You can see some green. So we are entering that third stage. So over the next 10 days or so, virtually the entire vineyard, this is all Syrah, will turn purple and will be kind of in that full phase three for that ripening process happening. So I did want to follow up with you. I did want to talk about some of the physiological processes of the grapevine. Now, please bear in mind we are going to keep this at a very high level. So kind of that 50,000 foot view, if you will, a look at photosynthesis and respiration in particular. Please know that there's an incredible amount of detail regarding these processes. Now, if you were doing plant biology or plant physiology, or certainly going to be a researcher in those spaces, you would need to know this in very excruciating detail. But for wine lovers, we don't need to do that. We're just trying to get an understanding of what the plant is doing. Just going to improve our knowledge of the grapevine a bit, so that way we can appreciate wine more. So first process we'll talk about is photosynthesis. At a very high level, what it is essentially doing is converting sunlight into plant food, right? And that plant food, the main photosynthate that it moves through the grapevine is sucrose, which is a 12 carbon sugar. So we are harnessing the energy from the photons and capturing that, and then using carbon dioxide that comes into the plant, and essentially using that energy to fix that into a sugar molecule. This C6H12 molecule, that is the formula for glucose, and that's kind of the simplest sugar that we have. Glucose and fructose have the exact same chemical formula. They have different spatial arrangements in 3D, so they are isomers of one another. So they are what we call C6 sugars, or 6 carbon sugars. The This is a very simplified equation of photosynthesis: 6 molecules of carbon dioxide, 6 molecules of water, sunlight, which is providing the energy, drives the reaction, resulting in 1 molecule of glucose and 6 molecules of oxygen. And this is thought to happen in two separate reactions. It was typically called the light reaction and the dark reaction. It was thought that the dark reaction didn't need sunlight to happen. I believe that has been proven to be false, but that name still kind of persists in textbooks. So I kind of put it in quotes here. So let's take a little bit deeper look. So here is a leaf cell, a leaf. We have where photosynthesis happens is in the chloroplasts. It's kind of a structure inside the leaf. That's where the light energy gets trapped initially. We have sunlight, light energy coming in. We have water coming into the leaf, which is picked up by the root system and kind of pushed up into the plant. The stomata, this structure on the leaves, allows for gas exchange. The carbon dioxide enters via the stomata, and in fact, water will also evaporate out of the stomata at times via this process called transpiration, which we'll talk about in a little bit. And then this energy is driving that process. The sugar gets generated, and then that will be transferred to various locations in the plant depending on the need. So the light reaction again takes place in the chloroplast. That initial light capture, that energy from the light capture is used to break apart two molecules of water into electrons and protons. H plus is hydrogen is one molecule. One proton, one electron. If we strip the electrons out, we have essentially a proton left, and the oxygen. So the oxygen that is the plant gives off oxygen, takes in carbon dioxide, gives out oxygen. The oxygen that's actually freed up and liberated back into the environment is actually from the water. And then it forms, also uses that energy. The energy is captured in the form of ATP and this NADPH. These are kind of the energy currency molecules of the cells. So that capture of energy is used to attach an inorganic phosphate to the ADP, producing an ATP molecule, so high energy here, and then also to attach a hydrogen on its NADP plus, kind of reduces that to NADPH. So these will later on be used to drive subsequent reactions in the carbon fixing phase to produce that sugar molecule. So the dark reaction, this is also known as Calvin cycle. Again, this was thought to not need sunlight initially, but I think it's been shown that it actually does need sunlight to function. So this is a harvesting using the energy capture in the form of ATP NADPH, and it is in a stepwise fashion going to convert the carbon dioxide to a glucose molecule. And one key thing that is needed is this enzyme called Rubisco. This is a ribulose 1,5-bisphosphate carboxylase, quite a mouthful there. It is the protein enzyme that is essential for carbon fixation. I believe, if you're ever on Jeopardy or anything like that, if they ask you what is the most prevalent enzyme in the world, I believe the correct answer would be Rubisco. That is, just think of all of the leaves that are out there, green leaves in nature. Then this is what's driving it. So it is the most prevalent enzyme, I believe, on the planet. So let's not get too bogged down here. So this is the Calvin cycle initial step. All of the red items here, the spheres here, represent a carbon molecule. So it takes three molecules of this compound, RUBP, this enzyme Rubisco, this RUBP carboxylase, 3 molecules of carbon dioxide, and it generates 6 molecules of a 3-carbon structure called 3-phosphoglycerate. And then it's going to continue using some of the ATPs that were generated, the NADPHs that were generated, to drive this down to glyceraldehyde 3-phosphate. 6 molecules here. 1 molecule of this is going to be shunted off. So this is a 3-carbon molecule. 1 of these will be shunted off, and 5 of these will continue on and regenerate Rubisco. Rubisco bisphosphate. So, and then the cycle will continue. So with each, so it's going to take 2 turns of this to take off 2 glyceraldehyde 3-phosphate molecules, and that since these are 3-carbon molecules, it takes 2 of them, and that's going to form a glucose, which is a 6-carbon molecule. Okay, so 2 turns of this cycle will generate 1 molecule of glucose. It can generate, but I did say the principal photosynthate that the grapevine will kind of move around is sucrose, and sucrose is a combination of 1 molecule of glucose and 1 molecule of fructose. So there are various enzymes that are present that could change glucose into fructose and vice versa, and then kind of combine them to make the sucrose. All right, so now that sucrose can go on, once it is at various places in the plant, it can be, through other chemical processes, be converted to proteins, fats, other carbohydrates, whatever what the plant needs. But that sucrose is that initial step. It is the foundation of everything, and is driven by the sunlight that comes in and the carbon dioxide and water to generate this. So plants can generate their own food. Now humans can't do that. We don't have any chlorophyll or things like that, so we can't do this process. The overall efficiency of photosynthesis is only about 6%. So not very, very efficient in the grand scheme of things. It's just that there's so many leaves and so much chlorophyll available. The plant doesn't really need to evolve to anything much more efficient than that. It can still function at a relatively low efficiency. Now, just by comparison, modern solar cells are approximately 30% efficient. Possibly that might have moved up to about 38% efficient recently. So it's much more efficient than photosynthesis in capturing that energy and being able to reuse that energy. So this only happens in green tissues. So where that's mostly the leaves on the stems and things like that. No photosynthesis really happening there. Okay, so so canopy management becomes quite crucial because we want to be able to any leaf that is in the dark, so to speak, no sunlight hitting it, is not able to do any photosynthesis. So it is crucial during the growth phase to open up the canopy so that we can maximize sunlight exposure hitting the leaves, and that will increase the photosynthesis rate in the plant. Some factors that affect photosynthesis. So the ideal temperature for photosynthesis is between 77 and 95 degrees. Anything above 95 degrees, the stomata on the plant close. And that's mostly a response because it's losing too much water at that time. So the stomata close. If the stomata close, there's no carbon dioxide getting into the plant. So essentially photosynthesis shuts down above 95 degrees. And if it's too cold, below 77 or so, there's not much photosynthesis happening because it's temperature dependent. The ideal, I believe, photosynthesis is optimized for the grapevine somewhere between 86 to 88 degrees, and that would depend on the specific clone and variety that you have planted. Again, if there's a lot of water stress, water deficit type of thing, the stomata will close and then photosynthesis is going to come to a halt. If you're in a severe nutrient deficiency state, photosynthesis is not going to run because that initial light capture is done via these photosynthesis complexes that need various nutrients in there to be able to be effective and formed. All right. So the respiration is kind of the flip side of photosynthesis. That is, now once this energy is formed in the form of sucrose, what can you do with it? Well, virtually all cells are able to do what is called cellular respiration. So it's be able to take a glucose molecule, some oxygen, and generate carbon dioxide, water, and energy. That energy is stored in the form of ATPs in the cell, and that can be used to drive and support any metabolic processes that the plant needs to do, or even humans need to do. Like all of our cells can metabolize glucose. So it needs oxygen for that to happen, right? So incidentally, in humans, if you run out of oxygen, like let's say you're working out really, really hard and you're pumping iron and stuff and doing a bunch of reps, at some point your muscles are going to stop burning. What's happening there is that the oxygen is getting depleted and it moves slightly into anaerobic respiration. And one of the things in human cells, and when it goes into anaerobic respiration, is to generate lactic acid. And lactic acid is that's what that burning sensation is in your muscles if you're working too hard, and that burn is lactic acid buildup in humans. So in the grapevine, it is breaking down those sugars and generating ATP. So you can think of it very much, respiration is kind of the opposite process of photosynthesis in many ways. The major processes of respiration, there are three key phases: glycolysis, Krebs cycle, and oxidative phosphorylation. The oxidative phosphorylation, you may have heard it also referred to as the electron transport chain. Exact same thing. Now, I'm not going into any of these in major detail or anything like that. There are numerous great videos on YouTube on all of this stuff if you're really interested in the nitty-gritty. Like I said, not much interest for us on the wine appreciation side of things, or even the grapevine appreciation side of things. Know that this happens in virtually all plants. and all living tissues can do essentially respiration. So substrates, you can use carbohydrates, fats, amino acids, organic acids. All of this stuff can get broken down ultimately via numerous mechanisms into glucose. One of the things I do want to point out will be on the next slide when we talk about the decrease in acid as this now phase 2, that lag phase is essentially over. Verasion is starting, right? What that means is the grape seeds are now viable, right? So from the plant's perspective, you can theoretically take those grape seeds and plant them into the ground and under right conditions generate a new grapevine. So once that happens, the plant's motivation is to now make the berries very, very sweet, reduce the acidity in there. Hopefully, an animal will come and eat the berry. It keeps the seeds very, very bitter. Those compounds around the seeds and the tannin compounds, all of that, very, very bitter. In the hopes that it will spit out, the animals will spit out the seeds and a new plant will germinate. So let's see. So let's go to the next slide real quick. So here is the Krebs cycle portion, kind of that middle phase. And one of the key things I wanted to point out is that if you recall in a previous lecture, I mentioned that to make the grapes initially, when the seed's not viable, the vine makes the grape berries very, very unpleasant to eat. And it does that by pumping it full of acid, namely malic acid. All right, so it pumps it full of this malic, harsh-tasting acid, a lot of bitter compounds. It also adds to it tannin compounds, keeps the berries very, very firm. So if you were to munch on a berry early in the season, they're super sour, not very pleasant to eat. So what I wanted to point out, though, is now once the seeds are viable, so prior to verasion, the grapevine's using the sugar because we're not trying to accumulate, it's not trying to accumulate sugar into the berries just yet. So it uses sugar from the photosynthase to drive a lot of the metabolic activity, so via this respiration process. Now, once the seeds are viable, it wants to pump all that sugar into the berries to sweeten it up. So it can no longer use that for the respiration process. But in the Krebs cycle, malate, malic acid, right, kind of in ionic form here, is a key component in the Krebs cycle. So this can drive the process of the Krebs cycle and the oxidative phosphorylation step to generate the ATPs that could be used for subsequent metabolic processes. So this is why at this stage, verasion onward, the acidity in the grapes starts dropping naturally because malate is now used as the energy source because it's trying to preserve the sugar to sweeten up the berries. Okay, so that's what's happening when the malic acid is dropping naturally in the grape juice. If you were to measure this, you'll see malic acid steadily declining from verasion onwards to harvest time. It may start off at 5-6 grams per liter. By the time you harvest it in end of September, October, that may have dropped down to 1.5 to 2 grams per liter of malic acid in there. And this is what's kind of going on. This process keeps going, right? The plant needs energy and in the form of ATP, right? So, in a very, very real sense, these are kind of complementary processes. Photosynthesis produces sugars from energy. Respiration, we kind of burn the sugars to generate energy. Energy is stored in photosynthesis. Energy is released in respiration, etc. This occurs only in the chloroplast. Respiration occurs in most cells in our body. The initial glycolysis step happens in the cytosol, and then the subsequent steps. The Krebs cycle and the oxidative phosphorylation happen in the mitochondria. Oxygen is produced in respiration. Is used in respiration because at the end of the electron transport chain in oxidative phosphorylation, oxygen is the final recipient of those electrons. Carbon dioxide gets used in photosynthesis. Carbon dioxide is produced in respiration, etc. So these are the key things that are happening. So as a result of this, one of the cool things is carbon dioxide is taken into the plant, generated into sugars, and it's releasing oxygen that humans desperately need to breathe. So it's kind of this regenerating type of thing. So we need plants to kind of regenerate that oxygen for us. So one other key thing that's happening is, okay, these photosynthase are being developed. The sugar is being developed. Where does it go? Well, it kind of goes wherever the plant needs. Initially, or a good portion of that sugars will be used for energy until veraison hits. Then it will move from the leaf to the berry. It'll get translocated there. Now you've heard, probably have heard xylem and phloem. For xylem, you can think of as kind of a one-way conduit for water and minerals that is taken up from the root system and moved through the plant. It's not really a two-way street. The xylem, the phloem is a two-way street, as it can go in bidirectionally, depending on the need. And this is where the photosynthase and other various compounds that the plant generates can be moved from one place to another in the grapevine. These are all kind of pressure-dependent type of activities, loading and unloading, if you will, type of things that are happening. And the common terminology in the textbooks will be kind of source versus sink. The source is where the photosynthase are produced or where they're stored, and the sink is where they need to go and be offloaded. So if you want to think of it, in the beginning of the growing season, like bud burst, and at that stage, there are no leaves on the vine yet, right? So the photosynthesis is not happening at all. So what is the energy what is providing the energy to continue these metabolic activities? Well, in the fall, the grapevine knows winter's coming. It stores the photosynthase in the root system and in the trunk and parts of the cordon. And once the weather starts warming up in the spring, it moves those photosynthase, those sugars out, and those are broken down via respiration to provide the energy to do the metabolic activities in the springtime, because there's no leaves on the vine yet, right? So once the leaves start emerging and they're able to do photosynthesis, a lot of that sugar is used to drive that activity in the beginning. At veraison time, that switches. The sugar is kind of put into the berry, into the root system for future use, and malic acid becomes the substrate that's used in the respiration process. So this translocation, the plant needs to be able to move these compounds throughout the vine, and it uses the xylem and phloem to do that. Xylem is mostly for water, and the phloem is mostly used for the photosynthase and compounds in the grapevine. And lastly, the other key process that is always kind of happening is this transpiration. So the stomata, those structures on the leaf that let in the carbon dioxide, they also let water escape through the plant. So the water, this transpirational pull, this kind of passive movement of water from the root system to the leaves, to the outside of the leaves, this is what's happening. Now, when the water, if the plant is losing too much water, like the temperatures are 90, 95 degrees plus, plant's going to start losing a lot of water. That evaporative loss that's happening off the leaves is going to be too much and at a too high rate. Interestingly, transpiration is what the primary mechanism is to cool the plant down during hot weather. But if it starts losing too much water, the stomata will close because it wants to conserve the water. Once the stomata close above 95 degrees, the stomata pretty much are closed on the grapevine. So even in the midst of all this beautiful sunshine, if it's over 95 degrees, there is no photosynthesis happening because the stomata have shut as a response to stop and prevent major water loss. But once the stomata are closed, their carbon dioxide is not being able to get into the grapevine. So the photosynthesis process will stop very, very quickly. These are the major processes that are happening in the grapevine, kind of running 24/7 almost. In the case of respiration. One other point I like to make is that it is temperature sensitive. So below about 70 degrees, the respiration process kind of slows down significantly, and this is why that we often talk about the diurnal shift in growing regions. Why it's so important, because that cooling aspect of temperature breaks or reduces greatly that cycle of respiration, so that malic acid gets preserved. In very hot climates, like in Texas or more equatorial type of climates, that respiration cycle doesn't break, and you get grapes at harvest that are very, very low in acid. So then the resulting wines will not have balance. So they need to supplement it by adding a lot of tartaric acid at the fermentation bin, or even afterwards, to get some acidity built back into the wine, to have a properly balanced wine at the end of it. So I think this is a good stopping point. Like I said, this was just meant as a very, very high level introduction into these physiological processes of the grapevine. There is so much more detail. Now, if you were a plant biologist, plant physiologist, but of course you would need to learn all of these in excruciating detail. But I think this kind of serves for our purposes what we need, just to get some appreciation of what the grapevine is doing, and kind of the winemaking process. So we'll continue tracking veraison, and we'll talk about in the upcoming videos what's happening between veraison and harvest. Really cool things are happening now, and this is where it's going to develop all the flavor compounds and all of that stuff coming up. So I'll be back with another video shortly with kind of explaining those processes in a little bit more detail. All right, thank you. Bye bye.